Alright, guys. Let's do this. How are we doing? Is anybody here? Oh, concurrent viewers, 1. Oh, we're at 1 viewer. Okay. 2 viewers. Let's see. Four viewers. Hey, Aziz, how's it been? Okay, I'm gonna wait for people to trickle in. Took a little bit of time to get everything set up. How fam? Hi, I'm good. How are you? Hi, Andrea. Hi, Mike. Hi, Vinesh. Hi, Vinesh. I saw your email. I promise I'm gonna respond to it. Okay. Can you guys hear me? Everything okay? Well, people. I guess we'll give people 5 minutes to come in. Hi, Samuel. Hi, Casey. Hi, Shada. Micah, do you have the cure for COVID-19? No, we don't have the cure yet, but we're talking about doing some computational stuff with it. Yeah, there's definitely a strong lag between the stream and me, but that's okay. Let's see, so it is 2:11. We'll start at 2:15. Give time for everybody to come in. Has anybody had anything cool happen to them in the past couple weeks? Anything you want to share with the rest of the 25 people who are here? Is today's lecture hard? No. No, I wouldn't say today's lecture is hard. Um, it might be a little bit confusing at first. However, you should be fine. I think you should be fine. I am very, 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 very busy. Are you gonna find the cure, Dr. Delaflor? Alright, we'll give people 2 more minutes. That's what I have been doing. LOL. That's good. That's good. At least we're gonna get a cure from somewhere. Oh, for those of you who did not hear, the Senate and the House just passed that really big stimulus package. $2 trillion for $2 trillion but somehow there's $6 trillion mixed in there. I'm not exactly sure on the details. I just know that most of us are going to be getting $1,200 checks, which is nice. Very nice. Free monies. Yes, free monies. Oh. Okay, one more minute. One more minute. Okay, we have 37 people in here. 39, about the size of a class itself. For those of you who have group mates who aren't on, go ahead and if you've been claimed as a dependent in the last 2 years and you don't qualify for the free monies. Oh, Casey, that's really important. Thankfully, I have not been claimed as a dependent, so we're all good. Oof. Actually, I think that'd be good for your family, right? Maybe? Because if they claimed you as their dependent, right? Then that means they get extra money for that, I believe. From what I've read. Okay, it's 2:15, 2:16. Let's see. We'll go ahead and start this up. Let me know if you guys have any questions, if you have any comments, concerns. Let's see. Oh, it's unfortunate. Oh, wait, actually, so that I can see the chat, I can go ahead and just log in on my other computer. Because if I had the PowerPoint up on this window, I wouldn't be able to see the chat, and that wouldn't be cool. And just log in on my other computer. Because if I... Okay. Alright guys, Micah, do I play Animal Crossing? No. My fiance plays Animal Crossing, and I really want to get her that Animal Crossing game that just came out. However, I went to the store, and it's like $300 to get a Switch, right? Because I'm not into buying that little Switch Mini or whatever it is. And then the game itself is like $60. So, I mean, that's a lot of money. Alright, let's get this thing started. Let me make sure that I can see this on my screen. Let's see. Man, there is quite a generous lag. what I'm going to do is shrink down this box just a little bit. And then we'll come back here and pop it out. Alright, so today we're doing the bioinformatics portion of the course. I'm going to give the first lecture. Charles is going to give the second lecture. You can email us questions about either lecture. We both know all the information, so it'll be completely fine. Let's see. Yes, we will be uploading the PowerPoint we're presenting. Mike Delafleur, who is insert your name? Oh, insert your name is another TA that we got the information from a long time ago. So, for the past few months, before we had to leave school, right, we focused on phenotypic analysis, we focused on chromosomal and allelic inheritance, and hopefully by now, because you've all submitted writing assignment 3, you know that you should have your gene identity, what chromosome your gene is on, what your complementation crosses, or what your complementation stocks were. You should know if it's sex-linked or autosomal. You should know if it's dominant or recessive. You should be able to tell us all of that in a paper. All right? We've given you all that information if you did not already have it from the lab itself. If you haven't heard by now, we have completely cut out the molecular section of the course. It's gone. There is no more molecular section. The reason why we did that is because grading and writing wise, it would be a lot harder for you guys to understand the material and a lot harder for us to grade off of that misunderstanding of the material. So we just cut it out. You no longer have to do that. So we are jumping in our class down to DNA sequence analysis and genomics. That means that the amount of information you'll have to know for the test has decreased a little bit, but you are also kind of specialized on the information that we've taught you so far. Let's see. Okay, so the whole point of this is to find the region of your gene that would have been amplified by PCR during the molecular section of the course. Alright? You're going to take that portion of the gene that should have your mutation in there, right? And you're going, actually, yeah. You're going to compare it to the wild type sequence, find your mutation between the two sequences, and then you'll tell us in a paper, hey, this is where my mutation is. You'll verify on Flybase that your mutation is there, because Flybase already has where all of these mutations occur, right? It already knows exactly where it is. Just look at the gene page for your particular allele, and you should be able to find it there. Then you're going to determine what gene region was amplified, and you'll examine your putative amplicon DNA sequences. Alright. So what does it mean when we say determine what gene region was amplified? You're just going to give us the amplicon, basically the region between the two PCR primers, right? And Charles will actually go over how to get that region later on. I'll slightly cover it today, but he'll actually go through the step-by-step of it. Examine your putative amplicon DNA sequences. The way we're going to examine this is by using a program called BLAST. It's online, it's free for everybody. I'll hit on it a little bit later in the lecture. Basically all you do there, you compare one DNA sequence to another DNA sequence, right? And it's going to find the homology or the similarity between the two sequences. Right? So in your paper, in your final paper, you'll tell me about the homology between the two DNA sequences, and then you'll go on and from that DNA sequence that you know that your gene produces, you'll go to the protein and compare the protein sequence with another protein sequence from another organism using BLAST. Alright. Let's see. Micah, are you still grading WA2? Yes, I'm still grading WA2. Okay. Oh. So relatively short DNA fragments can be sequenced by the dideoxy chain termination method. Modified nucleotides called dideoxyribonucleotides, DDNTPs, are attached to synthesized DNA strands of different lengths. Each type of DDNTP is tagged with a distinct fluorescent label that identifies the nucleotide at the end of each DNA fragment. DNA sequence can be read from resulting spectrogram or chromatogram. I'm going to go into this a little bit more later on. This structure of a DDNTP, so a dideoxynucleotide triphosphate, you should know this structure, right? You should be able to identify the base. You should be able to identify the 3 phosphate groups, so the alpha, the beta, the gamma. You should also be able to identify this ribose group right here. Know what the 3 groups are called, and then also know why it's called dideoxy, right? So normally we call it deoxyribose because there's only 1 oxygen here. But now that we have dideoxy, right, you have taken away 2 oxygens right here, which means that you cannot continue polymerizing the DNA, right? So once one of these fluorescent probes bind, right here, so once one of these fluorescently labeled probes binds to your chopped up fragments, you can no longer continue the sequence, right? You can't keep on going. So, a diagram of it. You've got your DNA, or your template strand, right? You take your primer, you got your DNA polymerase, you have your deoxyribonucleotides. These are just they have 1 oxygen group right here. And then you've got your dideoxyribonucleotides that are fluorescently tagged, okay? So the important portion is that you have both of these, right? You take your DNA template strand and you let your dideoxyribonucleotides be polymerized onto there, and at some point during the polymerization, you're going to catch one of these fluorescently labeled tags, and boom, the DNA polymerase is not going to be able to add another deoxyribonuclease, and you're going to get something that looks like this. Alright, so up here we've got the 5' that starts with, starts with C, and then it goes all the way down to 3' that ends at A, right? And we've got our polymerase that slowly sequenced this. So it looks like in this one, it hit the fluorescently labeled tag immediately, right? First base on there was fluorescently labeled, can't do anything else with it. This one, CCG, CCGA, and then CGAAA, CGAAG, and it just goes on and on and on and on and on, right? The big thing that you take away from this is that every time you incorporate one of these dideoxyribonucleotides, the polymerase can no longer polymerize, and now you have a set in stone piece of DNA, and that's it, alright? The next thing you do, you've got all your DNA strands right here. You put it through a column, and this is just like gel electrophoresis, right? So the heavier strands of DNA are going to be higher up, and the lighter strands of DNA are going to be able to travel faster. And those lighter strands of DNA all the strands of DNA are going to have the dideoxyribonucleotides on them. The shorter strands are going to come out first. Okay, so as they separate on this column, you're able to see with a laser what the fluorescent labels are, right? And then you get this wonderful looking chromatogram right here. Boom, 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 nice sharp peaks. Oh, yeah, there we go. Okay, and then at the end, you get a nice chromatogram that looks like this, right? This is, it's pretty decent. It is pretty decent. So if you notice, like at the very beginning, you've got all this choppy stuff over here, choppy, 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 choppy. These are not good reads, right? So in an actual scenario, you wouldn't count these reads. You'd say, this isn't good enough. You'd throw them out, right? And then as we get down here, you've got some really choppy reads. Let's see, it's pretty choppy right around, we'll say like right here is when you probably cut it off, and this is the usable sequence. See how you have nice peaks, peak, 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 peak. Those are what you're going to want to use in your blast sequences, right? And we'll upload all of the sequences for you guys online. Let's see. Am I not seeing any of the comments? Oh no. Okay. Okay, yeah. Just no one's commenting. That's fine. That's fine. That's fine. All right. So, to analyze the sequences that we put online, we're going to provide you guys with Finch TV program. It'll just be a little link on Blackboard, or you can just type into Google Finch TV program, and you'll be able to look at the chromatograms that we give you. All right. So, let me just walk you through that real quick right now, just so it's nice and easy for you. Alright, so you go to finch.tv, or just type in Finch TV download for Mac. Let's hope I just didn't download a virus. Okay, okay. Okay, I think this is the website. Download Finch TV for Mac. There we go. Alright, and then you got Finch TV right here. Alright, so you've downloaded Finch TV. The next thing you're going to do is look up a DNA sequence, right? So I'll just grab one off my computer right here. And let's see. We'll give you these DNA sequences online through Blackboard. So we'll just click on one of these. And it pulls up a chromatogram, just like this. Okay? So right now my computer is automated to go through a different software. However, your chromatogram is going to look very, very similar to this. Alright? So you'll pull through it. You'll take whatever amount of DNA you'd like to take. Right? And then you just command copy on your computer, or control copy, and you control paste that into a Word document. Okay? So, we'll go back. And over here. Boom. Alright, so now you've got your Finch TV. You're able to open your DNA sequences. You're able to look at the sequences, figure out what reads you want to take and what reads you want to put through BLAST eventually. Right? And so now we're going to go back into the bioinformatics portion, and we're going to be using BLAST to analyze genes, genomes, the protein sequence, protein structure, and functions of the genes and proteins. Let's see. Also, we can't comment right now? Why can't you comment? That's sad. Let's see. That is quite an unfortunate thing that you can't comment. Huh. Okay. Wait, I'm seeing, yeah, people can comment. Are you guys able to comment, or let me know? Maybe only mine is weird? Yeah, maybe, George, maybe. Okay. You're commenting right now. Okay. Anyways, Okay, so numerous genome sequence projects have been established or have established databases and refined analytical software to make data available on the internet. Right, that is the wonderful thing about science. Almost everything is free in terms of the databases. There are some that are pay to play, um, and then like some journals make you pay to read their articles, which is kind of sad. But thankfully, at the University of Houston, we have subscriptions to most of them. All right. So bioinformatic resources are provided by a number of sources. The National Library of Medicine and the National Institutes of Health created the NCBI, or the National Center for Biotechnology Information. There's also the European Molecular Biology Laboratory and the DNA Databank of Japan. And all of those databases can be accessed by BLAST, which we're going to be talking about a little bit later on. Software is available that allows online visitors to search GeneBank for matches to specific DNA sequences, predicted protein sequences, common stretches of amino acids and proteins. All of that's going to be covered in BLAST. NCBI website also provides 3D views of all protein structures that have been determined. And actually, one of the things that I kind of like on the final paper is if you provide a protein structure of the protein that you guys are talking about. I do a lot of protein biology. I'm trying to find a structure of a complex right now, right, through cryo-EM. So if you guys want to put a protein structure at the end of the paper, even if it's not required, you can do that. That's cool. I like that. Just put a protein structure. You can color it however you want. They've got a lot of really cool colors. So if you want to do that, that's something you can do. Alright. So bioinformatics visualization. Your query is going to be what you input, and then you're going to get all the subjects down here, right? So this is what is lining up to whatever query you inputted. It looks like this is homologous in all of these species except for your query, where our query right here is an R. Anybody know what R is? Oh wait, dang it, there's such a lag that it wouldn't even make sense. Oh, that's sad. Okay, R is arginine, right? And then right here, it looks like they've got threonines that's conserved right here, a serine, an alanine, two valines, and it looks like our query has a serine, so similar to humans. And then right here, our query has a serine, but all of these are aspartic acid. And then right here, we have an H and a Q, a histidine, and a glutamine, and then we've got WD, WD, WD. So these 4 regions right here are regions that you probably want to look at on a protein structure to see how the protein structure differs because of those differences. Concept of sequence similarity searching. So one sequence by itself is not informative, right? When we first sequenced the first organism, right? So the first full genome sequence of any organism, we were like, great, awesome. We've got all this data, but unfortunately we don't have a lot to compare it to, right? So the big accomplishment was just getting that first sequence. But then we started to sequence more and more and more and more and more, and suddenly, boom, we're able to, I don't know, get pretty much any sequence you want, pop it into BLAST, and you get so many different homologs all the way through. It's very rare that you're going to find something that doesn't have a single homolog, right? And if you do, that's something that you may want to look at. It's a little bit harder to look at because there's no homologs, no previous information about it. However, if you think it has a very specific and important function, that could be a novel protein that could be targetable by a drug. Develop hypotheses, okay. Actually, there's something I want to tell you about. We're going to go into a little bit of story time here, right? So I was mentioning how one sequence couldn't really tell you much, right? So the first full genome sequence of anything, right? Not going to tell us much because we don't have much information about anything else, right? But over the years, we've been able to get the sequences of thousands and thousands and thousands and thousands and thousands of organisms, hundreds of thousands of millions of sequences on this database, right? And it actually really helped one time. So, long ago, not long ago, a couple years ago, right? Europe had an issue. They noticed that a whole bunch of people were getting sick, right? So a whole bunch of people were getting sick, and so some of the top scientists around the world were looking for an answer, right? So they looked for about a month or so, and they narrowed it down to a case of E. coli, right? May not have been E. coli, but a certain bacterial organism that was making people sick, okay? So they sequenced that genome, and they were like, okay, we think we have it. We think it's from some crops in Germany, and so we're not going to import that crop anymore. Great. So they stopped importing the crop, and they waited. They waited for a couple weeks, and they realized, oh shoot, nothing's happening. This problem's getting worse. What are we going to do? China was like, hey, guys, I got you. I'm going to do this whole genome sequencing on this sucker, and we're going to get this done, okay? So Europe was like, okay, fine, go ahead and do it. And they did the whole genome sequence. They mapped it back to an exact strain of the bacterial organism that they were dealing with, and they found that somebody else had recorded that strain in Egypt, right? And they realized that it was actually grain from Egypt that was coming over to Europe and were infecting people with that bacteria, right? So they stopped the transport of that grain from Egypt, and boom, there we go, problem solved. Right, so there's the power of sequencing and bioinformatic analysis. Okay, so Bioinformatics BLAST Basic Local Alignment Search Tool. Basic Local Alignment Search Tool. I'm going to say it again. Basic Local Alignment Search Tool. It's underlined and bolded because you probably are going to have to see that on the test sometime. Let's see. Oh, okay, so everybody can comment. Yeah, sorry, I'm kind of talking and looking at the comments at the same time. So what does our basic local alignment search tool, or BLAST, tell us? Or what does it do? Well, it's a computer algorithm that aligns two sequences locally. Okay, it aligns two sequences locally. BLAST is basically the algorithm that takes all the information from the different databases and does something with it. Okay, so we could have all of the databases in the world, but if we don't have an algorithm to analyze the data from those databases, we basically have like a whole bunch of useless information. Okay, and there's a couple variations of BLAST that are going to exist. Let's see. So, basically, how this works. Right, so you input your sequence, you submit your query sequence right here to the BLAST web page. And BLAST takes that query sequence, puts it over to the server, and takes the information from the other databases, right? It uses this algorithm to align your sequence with those sequences, and then it returns the results to you in this wonderfully colorful diagram right here, right? And see, back in the day, okay, give me a second here, because back in the day, BLAST was not pretty, okay? BLAST looked like one of those 404 not found URL pages, alright? It's come a long way in terms of usability, in terms of how it actually looks. So when you go and use BLAST for the 27, 30 times that you have to use BLAST for this class over the next couple semesters or the next couple of weeks, just think back in your head to those graphic designers who were designing the BLAST page and say thank you, because they're what makes your life a little bit easier. Okay, so I was talking about the different kinds of BLAST that you can use. BLAST-P and BLAST-N are mainly what we're going to be using in this class. However, you do or you should know what the other types of BLAST are. Right, so we're going to use BLAST-in to compare a nucleotide sequence to a nucleotide sequence database. We're going to use BLAST-P to compare an amino acid sequence to an amino acid sequence database. Right? Okay, so for the BLAST algorithm, for those of you who are panicking about what I'm about to tell you, don't. You're not going to need to know it in as detailed as we're explaining it today, and I promise you it can go 100 times more detailed for how to get a better algorithm to align DNA sequences and really, really long sequences. Right, so the basics is that sequences are split into words, right? So words as in, like, if you had a sequence of nine bases, right? AAA, GGG, CCC. The three words would be AAA, GGG, CCC, and then if you shift one base over, you'd start counting three again. 1, 1, 1, 2, 2, 2, 3, 3, 3, right? So the sequences are split into words, right? The reason why they do this is for speed and for computational efficiency. So instead of matching a whole sequence to another whole sequence, which might not give you anything, right? They take it as little words at a time, right? So if these little words match up to something, and then a word next to it matches up to it, and then they keep on matching it down the line, down the line, down the line, then they say, okay, so we probably have some homology here. BLAST algorithm extends the initial seed hit into a high-scoring pair, or an HSP. Know that HSP is called a high-scoring pair, right? So the BLAST algorithm extends the initial seed or hit into a high-scoring pair. Okay, statistics are important. We have to discriminate between real and artifactual matches, so just matches that come around just because it's a little bit similar, but it really has nothing to do with your protein or DNA sequence. It uses an estimate of probability that the match might occur by chance and produces S-scores and E-values. Okay, for your papers, you're going to give us the S-scores and the E-values of all of your BLAST results. Okay, so in your results section, when you talk about your BLAST results, BLAST-P and BLAST-in, you need to give us the S-values and the E-values. And in your discussion, you should talk about the cutoffs, which I'll talk about shortly. Okay, quality of each pairwise alignment is represented as a score, and the scores are ranked. Scoring matrices are used to calculate the score of the alignment base by base or amino acid by amino acid. Alignment score S will be the sum of scores for each position. Honestly, everything I just said, you don't need to know. Why do you not need to know it? Because this is the culmination of what I just said, and there are entire classes on how to do this, okay? It's not something that needs to be covered over a live stream. Actually, this brings up another interesting scenario. Going back to when I was an undergraduate, I took a class that basically went over the algorithm for BLAST, right? And by the way, if you want to tune out for about 30 seconds, you can. Just trying to provide some comic relief for those of you who have to sit through an hour-long video later. Right, so I went in this class, undergraduate, and honestly, I was not the best student in this class. My other classes, I was there, I was ready to go, but in this class, I learned that I could read a 300-page book in about 3 classes worth. Okay? So I read 9 books, and there were 9 classes. So, let's see. During the test, I didn't study at all for it. He came and gave us like this free response test. It was 10 questions. He was like, go, you have an hour. And I was like, okay, let's do this. I didn't study, haven't even taken this class, and basically, it was just a logic test, right? And for one question, I distinctly remember drawing a circle. Like a circle with that had the little arrows on it, kind of like a recycle sign, right? But it was just a circle. I got my test back. about 2 weeks later, and I got a 100. I have no idea how to this day. Still have no idea how I got a 100 on that test, but I did. Um, so yeah, you don't need to know this stuff right here. Okay, but you do, or you should understand what this is. This is actually important. If you want to put it on your fridge, maybe save it as a significant other's profile picture. That would be good. Because any of you who are going to do anything in biology or biochemistry in the future, especially, need to know this. These protein sequence similarities are taken into account, right? Whenever you are aligning protein sequences together. So if you were to align a sequence with a whole bunch of aspartic acids in it, right? What's very, very similar to aspartic acid? Oh, wait. I'm just kidding. You guys aren't on the chat. It's glutamic acid, right? So if you had a glutamic acid switched in for an aspartic acid in one of your sequences that you're aligning, you can still say, hey, this is pretty similar. It's a very similar amino acid in forms of the charge, right? In terms of polarity. So that's something else that the protein sequences take into account when they're trying to figure out the homologs between different sequences. Okay, so what does score and eValue really mean? This is something you should know. This is something that you should definitely know for the test, for the quiz, and also for writing your paper. Right, so the score is the sum of the substitution and gap scores. eValue is the significance of each alignment. So the lower the eValue, the more significant the score. Alright, the lower the eValue, the more significant the score. Low eValue suggests that the sequences are going to be homologous. It's difficult to show non-homology, right? Because think back to kind of like chi-squared, right? When you're doing a statistical test, you can tell me that two things are something, but it's really hard to tell me that they aren't something. Right? When we're doing chi-squared, we can never accept the null hypothesis. We can only fail to reject. Right? We can only fail to reject the null hypothesis. So that's something that you can kind of compare to when you're trying to wrap your head around this eValue. Okay, eValue increases as database gets bigger, and eValue decreases as alignments get longer. And remember, the lower the eValue, the more significant the score, right? So as you have longer alignments, which means you have more homology, your eValue is going to decrease. Let's see. Similarity can be indicative of homology. So if two sequences are significantly similar over the entire length, they are likely homologous. Right? Low complexity regions can be highly similar without being homologous. The reason why is because low complexity regions oftentimes fold into like a loop or a turn in a protein, right? And so if it's not like a beta sheet or an alpha helix, you could still have quite a bit of homology between the two. Homologous sequences are not similar. Okay. Ooh, this is important. You should know this for your paper. Please know this for your paper. Genomics and proteomics. Okay, yeah, know this for your paper. eValues, actually, Aislinn Sweeney asked genomics and proteomics question mark. I'm not sure what she meant by that. If you have another question, just specify a little bit more. So for nucleotide-based searches, eValues of 10 to the negative 6th or less, and a sequence identity of 70% or more. For protein-based searches, you want an eValue of 10 to the negative 3rd or less, and sequence identity of 25% or more. Okay? So for nucleotide, 10 to the negative 6th. For protein, 10 to the negative 3rd. Right? So if you had an eValue for a protein-based search, so you inserted the amino acid sequence, you got a whole bunch of different reads back, right? if your e-value is 10 to the negative second, you probably don't want to use that alignment. All right, if it's 10 to the negative fourth, that is a better alignment. Okay. This is kind of a repetition of what we saw earlier. Okay, so just kind of, this is a bit of an example as to what the program actually does when it's lining up your sequence with another sequence. All right, so our query right here is RDQ. All right, we look at RDQ. RDQ, they assigned a perfect value of 16. Okay, and then they changed it, RBQ. So they changed one amino acid in the middle. Then they did RDZ, one amino acid that they changed. KDQ, one amino acid that they changed. Okay? And notice that the scores are going down even though they're only changing one amino acid. The reason why is because B is probably more similar to D, right, than R is to K. Right? So see right here, they changed one R into a K, and it dropped 3 points, whereas they changed the D to a B and they dropped 2 points, which means that this R right here probably is more similar in the physical and chemical properties of that amino acid. Alrighty, so they've realized that this word right here, RDQ, they line it up with another sequence right here, REQ. Okay, so RRR, D plus E, QQQ. This plus sign right here means that they're similar. Right, so in my example before, I used aspartic acid and glutamic acid. The protein symbol for aspartic acid is D. The protein symbol for glutamic acid is E. Right, these two amino acids are very similar. Right, so they put a plus. It probably means that it still has the homology there, right? Protein homology. Okay, and then they extend outwards from that initial hit right there. They extend outwards. Boom, boom. And they get farther and farther out, and they realize that they're losing homology. So it's most likely that this domain of whatever protein you're working with is similar to whatever you're comparing it to. And Aislinn Sweeney said, genomic and proteomics for the class you took. No, I did not take genomic and proteomics, or at least that wasn't what it was called. It was like biological algorithms or something like that. I can't remember. If any of you have friends who went to A&M, maybe they took it. It's not a bad class. Honestly, I did learn some very cool things in there. However, I'm probably not going to be applying it for a while, I'd say. Maybe sometime in my life I'll regret it and have to go back and read all that algorithm information. Bioinformatics, extending the high-scoring segment pair, HSP. Right? So we talked about how you should have a minimum score, right? This is their minimum score, and anything below this score isn't going to be what we would use as a high-scoring pair. Anything above is what we would. Okay, this is another BLAST readout. A lot of homology here. This is actually something that you're going to be putting into your papers. Right, so this is a good example of what you would put into your papers. You would tell me the E value right here, the E value. Then you tell me the score. This would be in your results section. The E value and the score for your BLAST analysis for nucleotides and proteins. Okay? And then this whole diagram right here. So from this corner right here to this corner right here, that's the whole entire alignment. You would put that as a figure in your results section. Okay, you would put that, or sorry, not results section, your figure legend. Right, you would put that as a figure in your figure legend section. Let's see. BLAST can find surprising alignments. That's true. That's true. It's different than just pure chance. And because of those alignments, we're able to see weird similarities that imply evolutionary homology. Right, so a good example of that is Micrococcus luteus, which is an organism, bacterial organism that my lab works with, or one person in my lab works with. And he's trying to validate it as a safe or safer model to model dormancy in tuberculosis. Right? And for those of you who don't know, tuberculosis requires a BSL-3 level lab. We do not have a BSL-3 level lab here. They're very, very expensive, and most likely as undergraduates, it's very difficult to get into a BSL level 3 lab or work in one. I had the opportunity to work with a group of scientists who had a BSL-3 lab and had access to that, so it was really, really cool. If you guys ever get a chance to go into one of those, please do. It's definitely a once-in-a-lifetime journey if you're never going to do something like that again. But going back to the main point, you can find some surprising similarities that imply evolutionary homology between two different organisms based on their blast results. Okay, so we'll spend the next 2 to 3 weeks doing all of this bioinformatics stuff via online instruction, right? It's going to be really confusing, right? All right, so talking right to the camera right here, right to you guys, okay? Let me get some water. Man, honestly, I really miss teaching you guys. Like, actually doing it in class, there's a huge difference between doing it in class and doing it here, where I'm alone. There's literally nobody here. There was a custodian here. Her name's Nora. She's super nice, but that was it. That was it. There was nobody else. It's just me. So I'm alone, just looking at this webcam and reading your comments, and it's kind of sad. So please, if you have questions about this, please email me. Please. I will answer any and all questions. This is confusing to do in person. Okay, it's going to be even more confusing to do online. So please, if you have questions, email me. Please. And be as specific as possible so I can try to address your situation, whatever situation you're in. Okay. So, I'm going to go over this a little bit, but Charles is really going to get into the nitty-gritty of this, the actual doing this portion of the bioinformatics segment. Let's see. So we'll give you a forward primer sequence and a reverse primer sequence. If you guys have not already gotten them on Blackboard, they will be provided to you shortly within the next week or so. Okay? You'll copy and paste the sequence into Flybase Blast. So you'll copy your forward primer into Flybase Blast. Alright, boom. You put it in right here. Blast is right here. I don't know why it says, oh, it says ignore. Ah, Dr. Delafleur is telling you to ignore this thing right here. Yes, if there's ever a paywall you can jump over. So, you click on Blast right here. Boom. You input your primer sequence. You press Blast. You wait a little bit. Now, in this time, while you're waiting, sometimes it can take, I don't know, a minute or 2 minutes. The maximum amount of time I've seen take is like 3 or 4 minutes. If it's taking any more than that, then just redo it. Restart it. Maybe your browser's frozen. Maybe something's wrong with it. Normally, though, you can just let it sit for a while and it'll do its thing. Okay, in this time, as a graduate student who like inputs really large sequences, this is when I take a drink of water, maybe put my head on my desk. You can cry a little bit if you'd like. But by the time the sequence is done being analyzed, I'd wipe your tears, finish your water, and get on to it. Okay, so the results page will give you all of this wonderful stuff right here. This highest scoring one right here is probably the one you're going to want to choose. You can either click right here, or you can scroll down and look at the score right here. See this E-value? So this E-value is 0.000059. some of you will not have e-values that meet the threshold. Or, yeah, some of you will not have e-values that meet the threshold. Alright? Just give me the highest scoring e-value. There are very few of you who will have an issue like this. However, some of you will. You'll have the score right here. Boom. Oops. And then this is where your primer aligns in the Drosophila melanogaster genome. Okay? This is where your primer aligns in the Drosophila melanogaster genome. So you go to gbrowse, you click this right here, and then you plus on the details. Sometimes it takes a little bit of time to load. Right? It might take 2-3 minutes max. Right? You click on the plus on the details, and it should give you something exactly like this. Okay? Oop. This is where your high scoring pair is, so this is where your primer aligns. Alright? There's your primer, and what you're going to do next is, well, you can do one of two things. You can copy all of the information from this right here, or in my experience, it's just more helpful to download the entire gene that your primer annealed in. Right? So you just click on the gene, and then you're able to get the entire sequence in a FASTA file. Or you can press download the sequence file, and it'll download this entire file of everything here. Alright, so you get all this wonderful information. You take this, and then you use, or you start at number 12 on the BLAST protocol for PCR primers on Blackboard. Okay, now, what I just told you, all of this stuff right here on BLAST, there's an easier way to do it. So, let me figure that out for you real quick. There's a website, and I'm going to look it up really fast for you. Oh wait, somebody asked, do you guys have a set day for when Charles will go over to specifically, or how to specifically use BLAST? There's not a set day yet. It will be out in the very near future though. Okay, so alrighty. I cannot remember for the life of me the name of that website. However, if need be, I can have another live stream basically where I just upload it quick, show you how to do it really fast, and you can get your amplicon a lot faster that way. There are multiple ways of doing this. In my opinion, both ways are valid. Right? Both ways are valid. And actually, let me show you how to use BLAST right now. I'm seeing, oh Mike, you want to use BLAST too. But you can try using BLAST on your own. It's not difficult. The slide show you how to do it. Alright, let me show you how to use BLAST right now. Let's open up a window. And we'll go to BLAST. Alrighty. And I'll look on my desktop right here. I've got the AlphaX transcript. Alright, AlphaX is my best friend. Okay? AlphaX and I, that's an integrin. We work a lot together. Alright, we'll take all of this information, all this nucleotide information. Since we have nucleotide information right here, we're going to go to nucleotide blast. Alright, so here's nucleotide blast, and we just paste it right in, hopefully. Oh, okay, give me a second. Command Alt, Control copy, or Command copy, Command paste. Okay, there we go. Alrighty, and say I want to blast it against everything, right? I want to blast this against absolutely everything. We just go to nucleotide collection, we press blast. Alright, we'll wait for a little bit, chill a little bit. Alright, and it looks like we've got Homo sapiens integrin subunit alpha X. Perfect, it is Homo sapiens. It looks like we also have a couple, gorilla, okay, mandrillus, colobus, hylobates, okay. So we've got quite a lot of homology with other integrins, or other alpha X integrins in other organisms. That's cool, awesome. Alright, now, let me just show you an example of the fly-based blast. So we'll go to flybase.org, and the gene that I've been thinking about for a while now is yellow. Why? Because the last people who emailed me were of the yellow gene. So we'll go to yellow gene, and I want to grab this sequence right here. So we'll say get sequence. Oh wait, loading a little bit. We'll give it some time to load. Okay, here's my sequence. Perfect. Alright, let's grab this right here. Okay, so now, flybase. So, at this point, right now, I'm at this portion of the slides, right here. I go to blast. In your case, you would take your primer. In my case, I just grabbed any random portion off of the gene that I wanted to find. So I paste it right here, and then I'm going to blast it. Alright, I'm waiting. My job is queued. I'm sipping coffee, shedding tears, looking at the beautiful, beautiful day outside. I realized as I was walking through my fiance's apartment complex, right? So as a graduate student, I'm normally in the lab like 24/7. Now that I'm not in the lab 24/7, I really like long distance running. So I just finished a run, and I was walking through my fiance's apartment complex, and I saw a lizard jump off a branch and scurry away and enjoying its own lizard life. And I was like, wow, you have the coronavirus has no effect on you at all. You're just living your own lizard life out in the beautiful, beautiful air and the sun and the sky, and we're all freaking out over coronavirus. Yep, that was interesting. Okay, so after that wonderful realization, we're back with DMEL on the X chromosome, and our yellow gene is in fact on the X chromosome. Awesome. So we click right here, boom, it'll automatically bring you to the first query, right? If you don't want to do that, and instead you want to look at these values down here to look at your e-value, which is probably a smart thing to do, you look at your e-value and we see that it's 3.306 times 10 to the negative 15th, which is good because what was the cutoff for nucleotides for e-value? I'll give you a couple seconds to think. It was 10 to the negative 6th. And then what was the cutoff for proteins? Give you a couple more seconds. 10 to the negative 3rd. Alright, so is this less than 10 to the negative 6th? Yes, it is. Do not be confused by this 15 here. It is a negative 15. So negative 15 is less than negative 6. You may found it surprising that I'm emphasizing that. However, people have done it before. People have done it before. Okay? That's all I'm going to say. Alright, so you would take this right here, right, and you'd click Gbrowse. Gbrowse would bring us to this wonderful page, and you get your high scoring pair, and it looks like our gene is going to be this gene right here, which is yellow, the gene yellow. Whew. Sorry. Yep, this is the yellow gene. Yay! So it's exactly where we thought it was. You would take this sequence, and what you can do is you can take this entire sequence. Oh, yikes. That's too much, too much. You can take this entire sequence, right? And it'll have everything from here to here, right? If you want to go a little bit more, go that way, you can go that way. But I think the best thing to do would just be once you've found where your gene or where your primer hits, I would just take the gene itself, right? I click on the gene and then get sequence of the whole gene region, right? So get the sequence, and I take that entire sequence right there. Actually, that's way too big. Maybe not the gene region. Yeah, you can do the gene region. Can't kill you to go over. So I get the sequence of the gene region, and then align your primers on that gene region. Find where your primers line up, and you'll be able to get the amplicon. All right. Charles will go into that a little bit more. He'll actually find amplicons for you guys. So, I guess without further ado, does it matter if we use the BLAST website or use BLAST on Flybase? No, it does not matter. No. However, the Flybase one does provide a very specific layout, and it's only for D. melanogaster, right? So just in case you accidentally aligned it to something in humans, and you were really confused because your human gene is not in D. melanogaster, oh, that's why we use the Flybase BLAST. Just in case, you know? It just eliminates that one tiny piece of error that could happen. Alright, any other questions, comments, concerns? Anything else? How are we doing, guys? Hello? Well, okay. Actually, I'll wait for like 30 seconds because of the lag. Oh wow, this is about an hour long. Okay. Maybe I can just type it in chat. So it would be a better idea to use both to double check our work. No. No, it would not be a better idea to use both. It would be a worse idea to use both, actually. You can do it with both, and if you do it correctly, it will automatically work, right? If you do it from Flybase BLAST to begin with, you're fine. You have nothing to worry about, right? The only reason why you'd have to worry about anything on the normal BLAST website is because it'll pull from all the other organisms unless you set it to do just D. melanogaster. Alrighty, well then, I'll give you guys like maybe a minute more. Pull any questions, comments, concerns, life. Will you upload this PowerPoint slide set? Yes, this PowerPoint slide set will be uploaded. All the PowerPoints will be uploaded. This video will be uploaded to the website or to YouTube. Was there anything I wanted to tell you guys? I'm grading your WA2s and WA3s. I remember. So WA5, actually, all throughout the entire semester, I've given you guys reviews on all your papers, right? So WA1, WA2, WA3, I'll give you reviews on WA4. However, I will not give you any reviews for WA5. You cannot send me an entire paper for WA5 and have me review it. The reason why is because that was what WA1, 2, 3, and 4 were for. Right? This last paper is going to be the culmination of everything you've learned from all those reviews throughout the entire semester. Okay? You can ask me questions about the assignment. You can ask me, hey, how should I write this? Do you think it'd be better like this or better like this? Should I include this? Should I not include that? You can give me all sorts of different little things or little questions about the assignment, but unfortunately, you cannot send me the entire assignment. And actually, that reminds me, as a bonus, for those of you who have stuck around, so I mentioned that putting a figure of your protein into the PowerPoint or into your final paper is something that you might want to do, right? So for those of you who want to do that, let's see. All you have to do is get the protein name, right? So for me, I'll just do integrin alpha X, right? Or I guess abbreviation ITGAX. And then uniprot. Integrin alpha X uniprot. So enter the protein name and then uniprot afterwards. Boom, you take it, and it's most likely going to be your first result, because normally the human or the Homo sapiens are always the first result. But if you're going to give me a protein structure, I'd like to have it as a Homo sapien model. If it's only in D. melanogaster, that's fine as well. Let's see. So then you go down here, you go to structure, and you've got this wonderful protein structure. Now this is not the entire integrin alpha X. This is just a domain on alpha X. This is actually the alpha I domain. And what you can do here is we can make this full screen, and you can color it in a whole bunch of different ways. So let's see, I'd prefer if you just take the waters out, right? And then make the cartoon. So you go over here, chain ID, entity ID, element symbol, see how it changes colors. If you do element symbol, rainbow, so you've got a whole bunch of different rainbows in there. You got some nice colors. I don't know. This is something if you just want to do it extra at the very end of your paper. It's not graded, but I do like colorful art, right? I like colorful things. So yeah, if you want to do it, it's great to have. I'm a structural biologist. When I see this kind of stuff, I'm just like, oh, yes, yes. So let me see what you can give me. Okay. Well, without further ado, I hope you guys have a wonderful day. Bye-bye.